Hey guys, welcome to the Dr. G Show. This is episode 155. I was just thinking, I think we've done this for like 4 years now. Like, this is quite a while to be this show's going on. So I want to thank a lot of you guys for watching this. For those who've been around even all 4 years. Hey, there's Tim and Sherry. I think we're episode 155, and many of you guys might have caught me last night on ADHD Live with Scott Spradlin and Mariah Warden. They had me on. I'm gonna move the mic over here, I guess. They had me on last night to talk about ADHD and kind of food and chemicals and all that stuff too. So hey, Gina, and it was pretty exciting because being on their shows is a little different because I'm used to doing my show for the last 4 years, but it was very nice to kind of interact with them and kind of hear their thoughts and perspectives, and they apparently really enjoyed my perspective for things, so that was pretty awesome. So that's the ADHD Live program that you can find on Facebook, and we talked about ADHD and genetics, and so I kind of want to touch on that a little bit today, but also go a little bit further and talk a little bit more about the methyl tetrahydrofolate reductase pathway, or methylene tetrahydrofolate, and kind of what that really means, and then also like an epigenetic kind of protocol for how do we maybe try to reverse or fix or ameliorate that gene mutation, what do we do for treatment, all that kind of stuff. So tonight, again, if I talk about stuff that a little bit more kind of like medicalese or too detailed, just say, hey, what the hell are you talking about? So I'm going to try to be very concise with it, because with a lot of the biochemical pathology, biochemical physiology too, it can be kind of overwhelming sometimes. So if you need clarification, let me know, and I want to hear your thoughts and your concerns. Hey, Sarah. As we go through this. So tonight we're concentrating on ADHD, or attentive disorder, or just learning disorders, and we talked about and its relation to one specific gene mutation. And I think last time we talked about like a diet has a lot to do with that, but this is one of those things where patients are very hard to treat. So if you've tried everything with your kids or everything as an adult to try to fix that ADHD, and you keep coming up just kind of frustrated, then doing a simple genetic test and having us run the data through the back end, we can actually look at very specific gene mutations and say, does the symptoms match what's going on? Yeah, so Gina says MTH, well, it's FR, yeah. So methylene tetrahydrofolate reductase. And all it does is anything with an ACE at the end, like reductase, is an enzyme that breaks down the first thing. So this is a reducing enzyme that's taking a 5,10 methylene tetrahydrofolate and reducing it down to a 5-methylene tetrahydrofolate. So again, it sounds pretty impressive, but all it's doing is just kind of simplifying that little methyl group. So that pathway is so important, and we've talked about this before on the show. But if you look at like the homocysteine pathway, so if you think about this, you have a homocysteine, which if it's elevated, it can actually cause heart disease, strokes, all kinds of problems. So we want to reduce homocysteine that's produced in our body, and we do it through methyl B12. So that homocysteine with methyl B12 becomes methionine, which then is also used in regulating mood and behavior. And then through folate, it then converts to SAMe, or SAMe, which is S-acetylmethionine. And then that one regulates mood and behavior also. And then through folate, you can convert SAMe into phosphatidylserine, phosphatidylcholine, which again are very expensive supplements for like memory, brain function, mood, but you also just make it through the folate pathway. And then through B6, with that folate, we can convert SAMe into S-acetylhomocysteine. And then the S-acetylhomocysteine will convert back to homocysteine, but 50% of that gets reduced through B6 into glutathione, and then glutathione is your major detoxification pathway in your liver. So this one homocysteine pathway is really important for mood, behavior, brain function, memory, clarity, and detoxification, and cardiovascular disease. So many people that we now know that cholesterol doesn't cause heart attacks, but we know certain cardiovascular markers are profound in determining whether we're causing cardiovascular damage, and that's homocysteine, which we just talked about, and then cardio-C-reactive protein. So with homocysteine, 50% of that's reduced by methyl B12, 50% is regulated or downregulated through B6 or P5P, peroxidine biphosphate. So just basic, again, very basic B vitamins are controlling this overall how you feel. And then once you have methionine, it goes through the folate pathway, and folate converts to, not folic acid, because folic acid's man-made, it's toxic for the most part, but folate converts from that 5,10-methylene tetrahydrofolate through that methyl tetrahydrofolate reductase pathway into 5-methylene tetrahydrofolate, or 5-methylfolate, or methylfolate. That's what you see if you buy it as a supplement, right? And the reason that's so critical through that through that folate pathway is because it creates something called bioterapin, or BH4 or BH2. And the reason that is so critical, that folate pathway reducing and making bioterapin, is because that's how we convert. Let me make sure I say these correctly. I made a little drawing. For some reason this isn't working. That's not working. Oh, there we go. Okay. Well, that's not working. Let me pull this up a little bit different way here. This will hopefully help simplify it a little bit. Here we go. Okay, so the bioterapin pathway, it's really crazy name too, because it's BIOPTERIN cycle, right? So we take folate and make it into BH2 to BH4. And so then BH4 takes very basic amino acids that we eat in our food, from plants and animals, but mostly from plants too. And then you have like L-DOPA, so things like velvet bean, fava bean. It uses BH4 to make dopamine, and of course dopamine makes us feel doped and happy and relaxed, and that's why people do drugs to feel dope, and that's why people do upregulating things like skydiving to help do that, or eat wheat and dairy, which is morphine, or sugar, which is more addicting than crack cocaine, and that's because that dopamine makes us feel dope, right? So we need that methyl pathway to make the precursor to convert L-dopamine into dopamine once it crosses the blood-brain barrier. If you're really interested, then it goes to the substantia nigra and the mesencephalon. So dopamine can also convert to norepinephrine and epinephrine, which we typically think is fight or flight hormones or neurotransmitters. But again, in small amounts, it allows us to have a certain amount of stress and get stuff done. Fire on all cylinders, kick butt today, right? Now, too much epinephrine, norepinephrine, well, that then ends up causing anxiety, right? So that precursor allows that pathway. It also allows tyrosine to make tyramine, and also make, hey, there's Mariah, to make tyramine, but it also allows it to make thyroid hormone, which is just tyrosine with 3 iodines. And then that makes a chemical called octapamine. Now, octapamine is a very important byproduct of this whole folate metabolism through this pathway, because octopamine is the one that regulates mood and behavioral functions. So when you look at kids with ADHD, or you look at just people behavior-wise, let alone like the most extreme, like autistic, that octopamine pathway is the biggest driver of whether people are responding the right way, let's say, or the wrong way, or appropriate or inappropriate ways. And so it becomes a great way to regulate behavior and kind of responses, that stuff. Hey, Mariah. And then, of course, that BH4, that bioterpene, it creates phenylalanine, which is amino acid, into PEA, and then that regulates mood and behavior and all kinds of derivative functions. And then, most importantly, it takes tryptophan, that amino acid, and then bioterpene from that folate pathway, then makes 5-hydrotryptophan, which people take as antidepressants and for the gut and for other stuff, even though that's not really the solution, because studies show that there's no neurotransmitter dysfunction in your brain. Even WebMD says in 60 years of research, we now know that's not true. But 5-HTP is important because it converts into serotonin. So for bone growth, for your gut function, and for a little bit of brain function, that's very important. Oh, Gina says she's having a deja vu, tonight. Why is that? And then serotonin through SAMe, which again we created through B12, through the homocysteine pathway, then is additionally used to make melatonin. And melatonin is not sleep. Many people take melatonin. You should never take melatonin. It'll downregulate your own production. You never take hormones. You take the precursors that allow your body to make what it needs. But when the sun starts to settle, it makes a red hue, right? And then that goes to your brain and then triggers a response that says, hey, we should make some melatonin, start winding things down, and then we'll use gamma aminobutyric acid, to then go to sleep. So GABA puts you to sleep, melatonin kind of tucks you into bed, if that makes sense. So again, through that folate pathway, we have stress, so we'll say get up and go, and dopamine to feel good, and then we have thyroid function, and then we have octopamine, which is behavior and mood, and then we have PA, which is mood, and then we have precursor for sleep and for kind of gut function and bone and all that, all as this kind of outcome to this process that starts with this one specific gene. Okay, I'm trying to read what Lynn says. Is SAMe okay for sleep? Now, SAMe is perfectly fine. Don't take melatonin, but SAMe is a precursor to help make more melatonin to go to sleep. Yeah, that's perfectly fine. SAMe is a wonderful thing for mood and even like really bad allergies and stuff. You want to fix the problem, but short term, that stuff works really good. SAMe is just kind of expensive. It's a byproduct of B12, which is cheap, but SAMe is like $1 a pill. So. So I hope I'm explaining this stuff really well to you guys in a way that's kind of meaningful. So that homocysteine pathway, that's what's doing that. Now, the chromosome, so we're talking about chromosome mutations, or what are called SNP, single nucleotide polymorphisms. So if we back up and we say, well, every single cell in your body, you have 50 to 100 trillion cells in your body, constantly rapidly turning over. And so they're constantly replacing their old DNA with new DNA. And about 3 to 7 years, your whole entire body is a completely new body, right? So whoever you are in now is not the person you're going to be in, say, a year to 3 years to 10 years, right? From bone to brain cell to everything. But most of your cells turn over very rapidly, which means there's a constant rapid transcription and translation of that DNA into the next cells that are splitting from that. So with that being said, every cell, all those 50 to 100 trillion, each cell has its own DNA strand. And that DNA, if we unwind it all the way out, it's 6 feet, right? And then you have 2 strands of chromosomes half are passed from your mom, 23 of those, and half of them from your dad. And then that makes up who you are, right? So we have this hard code genetics, and then we also have what's called phenotyping genetics or epigenetics, which is the things you can really control. Can't change my eye color, but I can change my predisposition for heart disease, cancer, diabetes, all those things. So we take that 6 foot of DNA, and then we chop it down into segments called genes, and then those genes have a little thing called a round histone, and that histone then encodes for the next segment of those genes. So everything can be replaced and duplicated very, very quickly as cells turn over. Those genes then, on each chromosome, control different aspects of our body. So some of those genes are kind of fatal genes. So if you look at trisomy 21, if those get flipped around, then you have catastrophic changes happen. But most genes are multifactorial, and so it takes many genes to cause an outcome. That's why eugenetics is so hard, is because the idea that you could change one gene and make their eye change, or their height change, or whatever, or their weight change, that's not how that works. Most genes or most characteristics of the body are multi-gene faceted. So you have all kinds of lineage changes that take place. And then once you have that DNA, it's not always the same. So the DNA that's in your skin, that cell should be identical to in your eye versus your saliva versus your blood, but that's not true either. So every single cell has the ability to cause little mutations. And within those genes, those 20 to 25,000 genes, we have different pairs that bind together, and there's only 4. There is C and T, I gotta do this right, sorry. T and A that bind together. So it can be an A and a T, or a T and A, but it can't be TT or AA. And then you have a C and a G, or a G and a C, but never CC, never GG, right? So those are the way those bind together perfectly. So that way when we replicate it, all we're replicating and dividing DNA is C's and T's and G's and C's. T and A, C, G. That's probably easier. So it's very simple. DNA is really simple when you get down to it, right? But if you have, think of your zipper, perfect, right? But if you have a T bound to a C, then this is broken. It doesn't work like that. So now that gene no longer works. That's called a SNP, or a single nucleotide polymorphism. One of those little nucleotides is in the wrong place. And so it can be a TT, it can be a CC, GG, AA, CAG, TC, But so there's a limited number of reactions that you can have from that are abnormal, and some are more common than others. Now, that being said, you have this mutation, but there's also this thing called DNA polymerase. And DNA polymerase actually goes through your DNA, and it looks at that histone and says the next sequence should look like this, and it makes sure it looks like that. If it's different, or there's a SNP, or a nucleotide polymorphism, then it edits out that mutation and replaces that, say, TC with a TA. And so it makes it back to right. And your body does this at the rate of 10,000. Oh, I think the maids are coming in. The cleaning people. I can see them out there. What are they going to do? Oh, it's okay. All right. So DNA polymerase reads that histone, and then it dictates what the next set is, and it fixes that DNA for us, right? So it does this at the rate of 10,000 DNA mutation fixes per day per cell. So that's 50 trillion times 10,000. So your body's constantly repairing DNA at this exponential rate. So then that calls into question why. Oh, it's okay.
It's okay.
Yeah.
Hi. Cleaning? It's okay? No
Yes,
cleaning?
It's okay.
Trash?
No, it's okay.
Okay.
Gracias. So, it's nice having your office taken care of, you know? All right. So, 10,000. Oh, so you have these mutations that are occurring because we live in a very toxic environment. There's all these things that mess up our DNA, like especially benzene, styrene, BPA, polyurethane, artificial colors, flavors, scents, all these kind of things, especially for Americans. And so these are causing point mutations, and then your DNA polymerase is editing out those mutations. So we'll talk a little bit about research, but the other part we want to talk about is kind of like, what do we do to fix that? So we'll talk a little bit about what's called epigenetics, and can we help DNA polymerase edit out mutations and stop causing the problem while we're reversing the problem? And that's your best way to overall fix DNA problems. And our body does it every single day, every single person. So when you look at someone that has a persistent mutation, what are they doing to cause it, and why aren't they doing things to reverse it? And when we look at the way to reverse it, it's very simple. So it's not all that complicated, which is nice. So on tonight's episode, I want to talk a little bit about that MTHFR, which is my favorite gene of all because it sounds so inappropriate. So it's on chromosome 1 at position 3622, if you're keeping track. So at chromosome 1, that means the very first chromosome, this thing is like rolling and controlling from the very, very beginning. And again, that's where you see like methyl tetrahydrofolate reductase, where that gene actually regulates and controls things like reproduction. So you'll see studies where there's recurrent pregnancy loss, and that's because folate allows you to close the neurotube. So don't ever take embryology because it's a nightmare. But we start out as 2 cells that become 16 cells, which becomes a morula, which is a little ball, and then those cells create a trilaminar disc. So at one point, I think you saw this on probably Facebook at some point, but basically we start out as a mouth and an anus, right? That's it. We're just a tube. And then from that tube, we then create muscle and then bone, and then another layer creates our nerves and our skin, and then another layer creates our guts. And so those trilaminar discs then start to make a little person thing, and then eventually, if your genes are right, you become a little person. If not, you end up with webbed fingers and toes and a horn out of your head. And we can make anything with DNA because there's no human DNA, there's just DNA. So most of our DNA is off because it makes other things. So that's the scary part about embryology. You realize at a certain point in utero, a fish and a bird and you all look exactly the same. So hopefully, there's Jenna, hopefully the right DNA turns on the right time and you start dissolving those webbed fingers and you end up with fingers instead of flippers, right? You look at something like a chemical DDT, it made a bunch of flipper babies, right? And that's because it interfered with that metabolic pathway that turns off that DNA and starts dissolving fingers, making fingers, right? So chromosome 1 is huge, huge, huge. I mean, from the very beginning, chromosome 1 is the thing that kind of dictates fertility as well as how well you're functioning. And then with the mutation, that SNP, there's several different mutations you can have. So it's at 677. And so you can have a CC, which is most common. So like we said, can it be CC? No, it has to be CG, right? Or GC. So a CC is the most common mutation. The most detrimental one is a TT. So it should be TA or AT, but a TT mutation at chromosome 1 at 36.22 ends up messing up that gene to where it doesn't work as well. But like we said, all cells are individual, so you can have mutations in different places and it not be in the right place to cause a problem. So that's why you don't see a 100% problem with this. Or some people aren't symptomatic even if they have that TT mutation. And then something like CT, so a C677T that you'll see on these reports, it's like 50% of the population, right? So it's very common, but is it common and match the problem? So that's where we see things like treatment resistance. So we talked about on ADHD Live with Mariah and Scott, we talked about treatment resistant ADHD. These kids aren't responding to anything, the adults aren't responding to anything. What are we missing? Well, that mutation is going to make those people less likely to be able to do very simple treatment protocols. And then TT, the T677T, has a lower activity than CC or CT, and that's about 10% of the population. So about half the population have the mutation, and they potentially could have like 38%, I believe, decrease. But the TT, which is the worst one, is only about 10% of the population. So, and then again, they have to be symptomatic. So DNA is not always destiny. They have to have symptoms that match this. Then there's also a nucleotide at 1298 on that methyl tetrahydrofolate gene, and that can be problematic because if there's the most common is an A and a C with that, and so if you have A1298C and you have the 677T, well then they can have up to, hey, there's Andrea. So they can have up to about 70, I think, to 80% decrease in folate metabolism. So if you know that folate is a big part of fertility, that can dramatically impair a woman's ability to maintain pregnancy, let alone create a neurotube that's closed so they don't have spina bifida or the brain on the outside or a whole host of all of these neurological problems, but really a lot that the body just says, all right, shut it down, let's reabsorb this baby, and then let's start again when the folate's a little bit better. The only problem is it doesn't get better because they have this mutation that has to then be treated with methylfolate and some epigenetics and some preventive stuff. So I hope that makes sense. And the other things is, there's research, and so we're talking about ADHD a little bit, but the other thing is things like dementia. And dementia, there's two genes that have now, like Bredesen's research protocol or whatever, his research that said, it's a multifactorial approach to dementia is how you fix dementia, right? So could it be vascular, could it be nutritional, could it be a mutation, could it be neurological, could it be dietary? Well, basically just address it as all of those things is what the Bredesen protocol does, which is kind of what we do. But even if you look at like all the testing, they test for APOE4, well, that's a gene, and let me see, I think I wrote this down. Okay, yeah. So that gene can be have a SNP on there, right? That's at chromosome 19, position 13.32. So at chromosome 19, if you have a mutation there, well, then you can end up with an increase of plaquing in the arteries of the brain in places that will end up causing either frontal lobe dementia or memory dementia or maybe visual impairments, or of course cardiovascular disease. So, and even kind of plaquing in the nerves, right? So they can cause a dysfunction, inflammatory reaction there. But it's not just the APOE4. You also need the methyl tetrahydrofolate reductase mutation. So chromosome 1 and chromosome 19 together kind of creates a bigger problem for dementia. But again, both those things can be addressed. And especially with APOE4, I did some research on that, and we'll do a program about that if we... Did we do it? I think we already did that. We might do a refresher, but those genes get turned on for inflammation. So again, even if you have a mutation, they're great. There's other APOEs that you want on to dominate, because then those will help decrease the inflammatory process and then help prevent dementia or even reverse dementia, which we've seen with the research. Hey, there's Carrie from Australia. Down under. Unless you're an Australian, I think Australia's on the top and we're down under. So we talk. Schizophrenia is a big one too, because we said TT is the worst one, right? So that's the less likely one, 10% of the population. But TT is probably the most prolific with that. And sure enough, we see people with schizophrenic behaviors, and then they're of course hard to treat. But if they have the mutation, then you can dose them with a methylfolate or methylene tetrahydrofolate, and you might be able to see their symptoms improve dramatically, right? Just like we would expect with ADHD. So, dementia, schizophrenia, pregnancy loss, and then ADHD, and then of course anxiety and depression. Those are huge ones. And a lot of you guys probably know that I probably have told you before, but I do research at KU Medical School. I'm part of a great, phenomenal team. I feel like I'm the lowest person on that totem pole, but oh my gosh, there's some big people, big shoulders that I have to measure up to on our group. But one of the things that we're researching is that same pathway and its relationship to behavior, which is awesome. Like this kind of stuff should really change not only primary care, but as specialties big time. And so some are starting to use this, but not everybody. So that 1298 and the 677, those mutations, especially added together, can dramatically impair a person. Now, my friend, who's a psychologist, she keeps referring patients to me, and with the presupposition that she thinks they have this mutation. And I keep wanting her to be wrong, but it turns out, so far she's batting 100%. So the way that you figure out if you have this, so if you have any of these kind of issues, and especially, but not just ADHD or learning disorders, you can just do a basic genetic test. So we found out that from like Ancestry.com or 23andMe Ancestry or the Health One or any of the others, basically they test your whole DNA lineage. So they map out all 20 to 25,000 genes. But they only tell you this much. So like the heritage, they're only going to tell you this much ancestry. With the health stuff, they tell you about that much health stuff. But you have that much information that they're not telling you about. And that's because people freak out. So the FDA came down on them a couple years ago and said you can't be telling people about their DNA like too much, because people are freaking the hell out. Because they truly believe DNA is destiny. So before we've talked about DNA is, for a lot of things, is predisposition. So when you look at disease, heart disease, cancer, diabetes, macular degeneration, any of that kind of stuff, it is predisposition with DNA. It is not destiny. Now, clinical outcome with things like methylene tetrahydrofolate reductase, those mutations can be profoundly important in treatment and understanding what the hell's going on with you and why things just don't seem to work. So either way, any of those genetic tests that you do, you can come to us, and there's probably other people, but we download the raw DNA, and then we take that DNA and put it through another software that lets us look at all the raw data. And so we can look at all 25,000 genes, and then we can look at all the mutations. So typically out of like 25,000 genes, the average person has like 200 to 300 mutations, which is fairly normal, right? And then we can go through those and we can kind of cluster them up. So you have like heart disease, cancer, diabetes. Those are typical. You're an American. That's like every other person. So of course you have a predisposition for this. You're American, right? So good. Eat healthier, basically change what you eat, drink, breathe, rub on your skin, and how you stress. And then we should see less and less risk factor for that. And most of those are small risks. But of those genes, we can look at very, very symptomatic genes that really are profound. And so if you look at the methylene tetrahydrofolate reductase genes, or CYP genes that are made in the liver that control, or the COMT that is methyltransferase genes, which then turn on and off DNA, those things are critical. Like let's say Kevin. So they're very critical ones because they are so important for dramatically changing the effectiveness of someone's treatment plan, right? CYP genes dictate which supplements you put someone on, which ones you don't, or psych meds, that kind of stuff too, right? Yeah, Andrew knows. America, right? So Gina says she gets back on 21 pills in the morning again. Exactly. So you can test those genes, look at those, and then basically tailor your treatment to things that are more effective, which is awesome, right? Now, that being said, you can have the mutation and still not be symptomatic. So I have methyl tetrahydrofolate mutation. I can't remember what mine is. Maybe CC or something. It wasn't one of the detrimental ones necessarily, but even though there's 88 variations. But I don't have any of those symptoms. You know, I feel pretty good as long as I eat and drink and breathe and take care of myself and change the way that I stress and meditate and do mindfulness. It's like I'm pretty good, right? But someone else may not be. So then that gene becomes a critical part of their treatment process. And there's two ways that we go about when we treat those kind of pathways if they are suspect. And we said in the beginning, folate from food goes through this pathway, this enzyme, to make 5,10-methylene tetrahydrofolate into 5-methylene tetrahydrofolate. And then that becomes the bioactive thing that runs the homocysteine pathway and the bioterapin pathway for thyroid, brain, memory, mood, octopamine for behavior, dopamine, epinephrine, norepinephrine, phenylalanine, all these wonderful pathways, right? So, if that person is symptomatic, they can't seem to just get a grasp on how to get back to feeling normal, then we can simply just give them 5-methylene tetrahydrofolate or methylfolate. And we should see, if we get to a therapeutic dose, a change. Then they become more compliant with just life or with the other treatments like talk therapy or dialectical behavioral therapy, DBT, or cognitive behavioral therapy or outcome-based therapy. And that's what you want to see. So, and I'm very surprised that this is fairly new information, but we've had patients where they have children that are just off the chart, but as soon as they correct that, they test it and they're like, oh, yep, that makes sense. That's probably what's causing this problem. They then treat that with methylfolate at therapeutic doses, and then suddenly the kid's like, okay, I can obey, I can respond, I can react, I can sit still, I can listen, I can read. And they're not freaking the hell out, right? And then driving everybody nuts and driving themselves nuts. They can actually feel like they are now in control. So that's a wonderful adjunct, a wonderful ability to kind of look at the back end of your DNA and say, is this an issue? And then have some therapeutic treatment for that. Like I said, that psychologist friend I have, she's kicking butt on that. So I thought the other day she messed up, but then I was looking at the wrong thing. And so then I just looked a different way, found the mutation. I was like, damn it, she's still 100% correct. So she's got a really good eye for noticing that as a psychologist of, hey, this person kind of, there's something about this that makes me think there's something else interfering with the process. So, excellent stuff. In that test, you can get really expensive versions, but the reality is if you get that done, it can be anywhere from $50 to $200. So again, very cost effective. It used to be like $3,000 to get your DNA tested. But then we can look at all these other things, all these other factors. So all the other genes and figure out really, are there other comorbidities coming on with your DNA? Yeah. So, hey Geraldine. So then the last part of this is how do we then, the part we're missing, and I have to figure this out. I'm going to try this for maybe either research that we're doing now or another research project, but figure out which things actually mutate that specific gene. So we know if it's chromosome 12 for leukemia. We know that benzene and benzene derivative chemicals cause a mutation in the chromosome 12 causing leukemia, right? So that's a no-brainer. We know those things. It's a matter of, hey, all these things that you eat, drink, and breathe, rub on your skin that are all made from benzene for greater profitability, you got to stop doing that, right? And the other part then is, can we have greater effect on our DNA polymerase to edit out mutations? So turning on and off genes And I think I read these. I'm going to read these real quick. So there was a couple research articles I pulled down before we talk about kind of epigenetics for ADHD, attention deficit disorder, hyperactive disorder, inattentive, for methylene tetrahydrofolate reductase. So this one here is from the Progressive, and I read these yesterday on ADHD Live. But it's from the Neuropsychopharmacology, Biological Psychiatry Journal, 2019. And it says altered methylene tetrahydrofolate reductase gene, so that's our MTHFR, polymorphism, so again, C is not matching to a G, a T is not matching to an A, in mothers of children with attention deficit and hyperactive disorder. And at the very end, the conclusion was the most important result of this study was that maternal C677C, methyl tetrahydrofolate reductase gene polymorphisms, are significant risk factors for ADHD, which that means moms that are expecting need to start getting this test done, right? And then start treating for that. Now, usually they do this anyways because they do prenatals, but most prenatals are folic acid or folinic acid. It's not folate or methylfolate. So you can take prenatals, and it ain't going to help with this if you have the mutation. It has to be methylene tetrahydrofolate. So that becomes a big issue for women that are childbearing ages that want to not only maintain pregnancy, but not have neurotube defects, but then also make sure that the kid's brain in developing develops adequately too, with respect to that mutation. So that's pretty awesome. And again, it says in here, like a lot of moms get folate during pregnancy, but it's not going to help if they have that mutation as well. And then the other one was the Journal of Human Genetics, 2017, and it said components of the folate metabolic pathway and ADHD core traits. And in this one, it said this indicated that the folate homocysteine pathway gene, methyl tetrahydrofolate, variants may affect ADHD etiology through mild homocystinemia, which that's not, we'll talk about that, and vitamin B12 deficiency, factors that are associated with cognitive deficit. So same kind of thing. If kids have learning disorders or dyslexia, those kind of things, let alone ADHD, that pathway becomes very profound for them. So, what were we going to say? You guys have any questions? This is kind of a complicated topic, but usually you hear me rattling off stuff, and then we have like Holly or a guest, like, okay, what's the simple answer to this? You know. But I want to talk you'll probably notice I'm talking a lot calmer, because I'm trying to make sure to not overwhelm with this stuff. Because again, it's genetics, and it's for most docs even it's like deer in the headlight kind of topic for us, Let me look in here real quick. See if there's anything that we missed. Neurotube defects, we talked about that. Preeclampsia, fetal growth, preeclampsia, maternal thrombosis, so like thrombus and their clots, strokes. Those are all ones that research shows that there's a lot of evidence that says this contributes to those. So for especially moms, even if they don't have that, they need to be doing methylfolate. And then it says some people have the hyperhomocystinemia, which we talked about before. So we said that homocysteine is a risk factor for heart disease and stroke, and 50% is reduced by methyl B12, then converted from folate to SAMe, and then through that's right. And then also folate then converts it to phosphatidylserine and then through B6 it reduces 50% of the rest of the homocysteine. So there's, it should be like 5 on your test, not 15, 20. right? They let it go to 10 or 11. But really 5 is where it should be. That way you know your B12 is working, you know your folate's working, you know that your B6 is working. However, even though it says that, the reality is that your homocysteine can be artificially normal even if you have that folate mutation, simply through exogenous kind of supplementation of methyl B12. Like if you do a lot of methyl B12, you may make that homocysteine well within normal. So instead of 50% reduction, you're doing 70%. Great, but that's, again, it's not a very good marker for that. So there's a lot of controversy with that, because theoretically it should elevate it, but because of what people do in treatment-wise, they may not see that elevated. Okay, so approximately 50%. Okay, so here we go. 677T heterozygous, so it's only one copy from a parent, not the other parent, has about 50% enzyme activity. But A1298C hetero, so again from one parent, 60% enzyme activity. And then if they have both of those, they have 36% enzyme activity. So that's the big one. 677T, A1298C combined, even hetero, they end up having about 70% decrease in their function. So more ADHD, more infertility, more lost pregnancy, more neurotube defects, more schizophrenia, more dementia. But the A1298 homozygous one, so that's from both parents on both sides of the chromosome, that's a 50% reduction versus 60%. And then the C677T homozygous, that's the TT one that we talked about, which is only 10% of the population, that's about 22% enzyme activity. So that one by itself, if you get both copies from both parents, there's about an 80% decrease, 78% decrease in your folate. Those people are going to have profound symptomatology at that point. So, hey, Donna Roberts. So, off to epigenetics as we finish out this. So, homocysteine is not necessarily the right way to look at that. And especially because there's 88 mutations, but there's 4 main ones. And so whether they're hetero or homozygous, that makes a big difference. So with epigenetics, that's a whole different beast here, right? So with epigenetics, it's a process of 4 main functions. So you have methylation. So most people are aware, not most people, most docs in my field, right, are kind of excited about methylation, right? Because methyl groups at cysteine at the right place in the DNA or that gene turns that on. It's like a key turns on. Or for oncogenes or other things, it turns it off, right? So that's what you want. The right methylation in the right places at the right time. And so the complexity of this, you can have like localized hypomethylation and global hypermethylation. Like you can have it the opposite. So that in the beginning, I was just like, well, how the hell do we fix this? And someone finally said, just let the body fix it. Put the ingredients in, it'll fix it. So I was like, oh, okay. So I tried that. So in general, when it comes to methyl, is then eating these methyl groups. So that's where those B vitamins come into, like methylcobalamin, methyl B12, or methylene folate, or I'm trying to think of the rest of them. Anyways, wow, it's escaped my brain for some reason, but we'll talk about that. So things to avoid in general when it comes to that pathway with epigenetics is gluten. That's a huge one. MSG is another big one. Those proteins both cause big problems. Inflammatory fats, because again, we can turn on the inflammatory genes, so that methylation can turn on bad genes. And then one of the biggest contributors for, say, a few things. So there's histone modification. So those histones that then tell the DNA polymerase what the next set of DNA should look like for the chromosome, or sorry, for the gene. We can do things to modify those, right? So histone acetyl dimutase, that's not right. Anyways, HADs, they're histone modifiers, and that ends up being things like ubiquination, methylation, sulfurization. What else? I think there's one more. Histone, and then there's also microRNA. There's a really good book I bought years ago, and it's crazy expensive. It's a little book, but it's just solid data research, right? And that's where I created my first treatment protocol for this, and saw some pretty interesting things happen for patients. But sulfur-rich foods, for part of that histone modification, is going to be things like onions and garlic, but also cruciferous foods like brassicas. So cruciferous vegetables, broccoli, Brussels sprouts, cabbage, any of those crunchy ones that we don't like to eat, those are, when you eat broccoli and cauliflower, it literally is changing your DNA. The most epigenetic foods of those are like parsley, celery, like that doesn't seem like that big of a deal. Turmeric, red wine, horseradish, apples, garlic, onions, nuts, berries, blueberries, beans, organic tofu, green tea, eggs, Brazil nuts, liver. Those are all foods that actually change DNA. The other cruciferous ones we have are collard greens and kohlrabi and kale, bok choy, Chinese cabbage, Napa cabbage, rapeseed oil, but again, it needs to be organic. Rapini, turnip, rutabaga. So like those mixed green ones are changing your DNA, which is pretty freaking awesome. And arugula, daikon, if you guys like radishes, daikons are huge, long radishes. Maca, which is good for hormones, but it's also really good for your DNA. Radish, pepperweed, wasabi, but not dyed horseradish wasabi that you get at, like, Wasabi, the restaurant. Real wasabi is green horseradish that grows only in Japan. So watercress is also a good one. So when you eat those cruciferous foods, literally changes what DNA is turned on and which DNA is turned off, and how to fix that mutated DNA or the SNPs. Same thing with sulfur. Fermented foods, like, I don't know if you guys do fermented foods, but like lots of people do kombucha now. But kombucha's only been here since the 1990s, but it came from the mountains of China, and it's this symbiotic bacterium colony of yeast that grows there. And then when the Russians invaded, they took kombucha back to Russia. Then the Russians went to vacation to Europe, and then the Europeans started drinking it. And then in the 1990s, Europeans came over here, and then they stole all our genes and left us with kombucha. So kombucha is just fermented, typically green tea, but it doesn't have to have tea in there at all. It can have herbal tea like hibiscus, or it can just, like I make, I think I have 9 gallons brewing in my dining room, but there's no tea in them. It's just straight kombucha water, if you will. And that's perfectly fine. But that symbiotic colony bacterium yeast, you use kind of like friendship bread, so it's been passed over all these countries all over from that mountain. And then you have to have that to grow it, right? So that's really good. But also kimchi. Any guys like kimchi? So you gotta get kimchi without MSG, but kimchi is just very spicy fermented cabbage and garlic. Kefir water's fine, but not kefir milk. And then Bubby's pickles, Bubby's sauerkraut. Bubby means aunt in Yiddish or something like that. So that sauerkraut and even the pickles, you consume those for like $6, but then you have like $400 worth of juice left, because fermented foods and drinks have about 10 times the amount of good bacteria in them that a supplement does. So it's a limited amount. You need 750 to 1,000 species. You're not going to get that through a supplement anyways. Get that through life. But fermentation is very important. Most all countries have fermented foods. And the reason fermented is so important for your genetics is it creates B vitamins, which then make, creates good bacteria in there, which then creates B vitamins, and those B vitamins make things like butyrate, and butyrate then feeds DNA polymerase and edits out DNA mutations at the rate of 10,000 times per day per cell. So that's pretty good. But also, they make methyl groups, which then turn on and off DNA. So you don't think of that kombucha as regulating your DNA, but it really does. The same thing's true with green tea. The phenylalanine epigallic acid that's in green tea is what actually creates methylation changes in your DNA. So sipping that green tea versus black tea is going to give you cataracts and aging faster, but green tea changes your DNA. And of course, omega-3 rich foods, greens, we talked about like the cruciferous ones are the best. And then toxic ingredients to avoid. We talk about that all the time with patients, but we have these general ones, but we're going to do some research to figure out what the specific ones for that specific gene mutation is. And I have a little time before I get that done, but I'll get that done and I'll share it with you guys. So when it comes to methylation, so we talked about those foods in general, but let's talk about specific ones. So with acetylation, ubiquination, phosphorylation, and sulfurization, there's 3 genes that we want to affect with that. In general, we want to do more, but HDAC modifiers are things like garlic and fiber-rich food that makes butyrate, which we just talked about. So again, adding garlic or mirepoix to all our dishes, that makes a whole different beast when it comes to food as far as regulating DNA. So Gina says oyster and fish sauce, if it's fermented without crap in there, it's perfectly awesome. In fact, almost all your condiments should be fermented anyways. All those vinegars, they should not be dead vinegar. They should be a live vinegar, like apple cider vinegar. And you can actually put those in your condiments and make them living. So every time you use the mustard or ketchup or relish, you're literally improving your DNA, let alone making you healthier and happier, more energetic. So I don't know, some of the oyster sauce has MSG in there. Some fish sauce does too. I know at the Asian market, I get the fish sauce with the fish in there and the little tiny, like, what do they call it? Minnows in there, right? But they only have like 3 or 4 ingredients. That's all you want, right? So the HDAC gene's pretty easy because that's garlic and fiber-rich foods or brassicas. The SIRT gene is modulated through resveratrol, which is red wine, butylene, which is fiber-rich food, quercetin, which is onions and apples again, lutein, which is or lutein, which is brightly colored foods. So that basically ends up being red grapes, red wine, apples, onions, nuts, berries, sweet red peppers, dark colored foods, celery, parsley. And then the last one is HAT, or the HAT gene, right? Curcumin and garlic and green tea are your main ones for that. And think about that. I mean, when we talk about these foods, and you look at like the expectations for America is 1 in 1 in cancer, let alone we have 10% with ADHD, we basically guarantee that you're going to have these mutations, and we're not going to eat the things that reverse and restore normal physiology, normal genetics, normal epigenetics. And so it's not rocket science when it comes down to this. I mean, the book that I use for this is like incredibly scientific. Like it often has more pages for the resources than they had explaining the damn process in the first place. But that means that other countries have an easier time at this because that's just their natural diet. But for us, we're going to look at this very complicated way to then force you, or kind of like nudge you in the right direction, to eat real fruits, vegetables, nuts, seeds, berry, legumes, and greens, and brightly colored foods, and green tea, and red wine, the things that most countries just take for granted. So, what else on here? I think that's it. And we're out of time, so that's perfect. So, in our process that we look at with this, looks like this. So that's the complexity, but it's really fairly simple, like most things. The science behind it, we can make it complicated, but the simplicity of clinical applications kind of just should be easy. So the other thing is miRNA, but that's affected through very much of the same foods. There's a few differences there, but overall it's basically the same stuff. So I hope you guys enjoyed this. This was probably the most complicated lecture we did. We don't have Holly or a guest here to slow me down and decomplicate things, so I tried to reel it in and go at a slower pace. But if you guys have questions, please ask those. If you have other topics that we want to talk about too, definitely post those. And I'll look forward to seeing you guys again. And for those of you that watch this offline, still post. I'll try to answer questions if I can. If I don't answer your question, don't tag me, right? Tag me and I'll get to it. All right. So sometimes things get a little bit behind. But other than that, thank you guys for joining me tonight. This is our biggest nerd session today, and this is episode 155 of the Dr. G Show. And thank you guys. All right. Bye, y'all.